I think maybe I can now. I'm recording now. So whatever I did worked. Okay, apologies for that slight issue with everybody. All right. Mihi ana ki te iwi me te whenua o Waikato. Ko Ange tōku ingoa. Hello everybody and welcome to today's seminar. I am going to start off just by introducing our speaker today. So Dr. Erin Hahn is a research scientist at CSIRO in Australia, where she's based at the Australian National Wildlife Collection. Prior to starting as the research scientist there, she was a postdoc at CSIRO as well. And then a little bit prior to that, she was also based at Australian National University across the road, which is where we met, and have known each other for a few years since. Okay, I think I'm just joining again because I pressed connecting to Zoom too many times. Anyway, prior to starting coming to Australia, Erin completed a PhD at the University of Arizona, and she sort of has a background in looking at temporal population genetics and epigenetics throughout this time. But in addition to that, Erin's a really excellent science communicator, as we're going to hear today. And she's also one of the Australia Superstar of STEM alumni. So I'll let you take it away from here, Erin. We'll look forward to hearing your talk today.
And thanks, Ange. Let's make sure I can actually share here. How are we going? How's that?
Yep, we can see that.
All right, I just want that. Hold on. There we go. Just I can see everyone's faces instead of my slides, and that's no good. I know what my slides look like. So long as they're not blocking for you guys, that's probably okay. So hello, everyone. Thank you so much for giving me the opportunity to talk to you today. I was really hoping to be there in person, but life sort of got in the way. So I'm glad to be able to at least be giving this talk virtually. Today, I'm going to give you a bit of a whirlwind tour of the sort of work that I do using degraded DNA, much of it extracted from historical specimens to gather as much information as I can about our changing planet. But before I dig in, I'd like to say that I'm going to be talking a bit about old museum specimens for a good portion of this talk. And I want to emphasize that old is relative. Tens of thousands of years before the first CSIRO scientists began their work here where I'm sitting in the shadow of Black Mountain, Australia's first scientists called this place by a different name, Gallenberry. And while the data that I'm going to be presenting today is old by genetic standards, it barely scratches the surface of the knowledge that First Nations people have about our planet, its environment, and the cosmos. So I'd like to acknowledge that I'm joining you today from unceded territory traditionally owned by the Ngunnawal and Ngambri people. And I'd like to acknowledge their enduring connection to land, water, and community upon which I live, work, and play, and pay my respects to their elders past, present, and emerging, and also extend that respect to the Waikato Tanui and any First Nations people that we have joining us today. As Ange mentioned, I'm at the CSIRO and I work in the the National Research Collections of Australia, and we house over 15 million specimens collected over the last 150 years or so, spanning vertebrates, insects, plant, fungi. And I would like to acknowledge also that many of these specimens were collected without consultation with traditional owners and in ways that disenfranchised and exploited those communities. This is something that our collections are in the process of acknowledging more openly and beginning to try to reconcile. And so I encourage you to reach out if you have any questions regarding that very critical work. And while my work spans many different taxa, the work that I primarily conduct is in the Australian National Wildlife Collection, where we primarily focus on reptiles, amphibians, birds, and mammals, and we preserve them in a whole bunch of different ways, both dried as puppets or skins, as well as we have a big egg collection, which many of it was donated to us by people who were conducting egg collections illegally. We also have a bone and skull vault, and the place where I spend a lot of my time is in the spirit vault, which is housing specimens that were preserved with formaldehyde and then popped into ethanol for long-term storage. I'm a geneticist, and so when I work in the collections, the sort of data that I'm focused on is their DNA. And the field of museum curation has been having a big boom in genetic research, and more we're able to use these specimens to not just gain little snippets of DNA, but we're also being able to reconstruct whole genomes from a whole variety of museum specimens preserved in different ways. But a whole wing of the collection has been largely inaccessible to genetic research, and that's those specimens preserved in formalin. The information, if we were able to get any of it whatsoever out of these specimens, was highly fragmented. And so most museums considered those collections completely off limits to genetic work and wouldn't allow sampling. And this is very unfortunate for researchers who want to get historical genetic information from taxa like reptiles, amphibians, and fish, because it's these specimens that are most frequently preserved in our wet collections. So they were almost completely lacking in historical genetic information. At the beginning of my postdoc here at CSIRO, almost 4 or 5 years ago now, I set out to kind of break open the vaults and give access to these sorts of specimens so that we can make the most of the specimens that we have in informing about historical change. The methods that I developed for extracting DNA from formalin-preserved specimens start at designing a system for kind of predicting the suitability of the specimen for sequencing so that we can limit how much destructive sampling we do of specimens unnecessarily, because we already have a good idea that they're not going to produce usable DNA. I then went and tested a bunch of different methods that had been previously used both in museum specimens as well as formalin-fixed and paraffin-embedded histological samples, and came up with a method that works really well on specimens that are of moderate or good preservation quality, even if they've been exposed to formalin. And it involves a very high-tech method of popping it into a pH 13 buffer, putting it in the autoclave for about 15 minutes, and then doing an old-school phenylchloroform extraction. We then do very heavy sequencing, followed by a trimming-free alignment, which maximizes the amount of endogenous DNA that we are mapping to our reference genome. And using this method, I've been able to recover DNA that will give genomic coverage of specimens upwards of 25x, which is starting to get to the point of the sort of cover that you would get from a fresh sample. So no longer is formalin a barrier to whole genome sequencing of historical specimens. I've applied this method to specimens as old as 1905, and with an eastern water dragon, got up about around 11x cover, and with some teeny tiny tuna larvae, which were so small that you could pick them up in a pipette tip. I was able to get 25x cover from about 45 or so individuals. And this method has now been published since about last year, and we've had a lot of uptake from it from curators and researchers around the world using this method so that we can have more reliable sequencing of these kind of specimens. And if you're interested, I've popped up the QR code to that paper for easy access. Included in that paper is, like I said, a decision-making system of determining whether or not a given specimen is going to be suitable for sequencing. And this is something that can be used by both curators and researchers in order to determine whether or not it's really worth it to sample that individual and conduct sequencing. And it can be done without disturbing the specimen at all. Essentially, you just look at it and you do what we call the ick test. If the guts are discernible and you can tell the organs from one another, there's a pretty good chance that it's going to be sequenceable. And then we just do a test of the specimen media, make sure that it doesn't have really low pH or extremely high formaldehyde, which might indicate that it was preserved using methods that weren't particularly ideal for sequencing. And then kind of depending on how much DNA you get out of the specimen will determine what sort of success rate you would have given your particular genomic output. So if you're trying to do whole genome sequencing, you'd want to limit to those specimens that you get the most DNA out of. But pretty much anything that yields more than like 20 nanograms of DNA, we've had really good success trying to amplify very small fragments. So you can do some kind of specimen ID at a minimum with most samples. So moving on from sequencing individual specimens, I also had at the same time a small kind of side project, which I affectionately call Bucket-Seq, which you'll see for reasons in just a moment. I know that I can sequence the specimens in the jar themselves, and this got me thinking, what else is floating around in this jar, in the specimen media itself? Because if I can use the tissue to sequence the individuals, then perhaps also DNA shed by those individuals or by their associated parasites and microbes, I would also be able to pick that up in the specimen media and sequence that and get an idea of the whole ecosystem of organisms that are preserved in that jar. So I went digging in the collections, literally in buckets in our prep lab, in order to test whether or not we can use specimen media itself for sequencing. And the workflow that I developed starts with those buckets of specimen media, and we apply a very eDNA-like approach, where I filter off the specimen media and then put that filter into an extraction buffer, the same kind of extraction buffer that I use for my tissue extraction, so that really high pH, and pop it into the autoclave. And so that contends with any potential formaldehyde preservation that the media has been through. And then I apply a metabarcoding approach. And so in this particular assay, I was looking at the 12S cytochrome and cytochrome B loci, as well as a 16S rRNA locus, which was designed to target some flies that we thought were potentially associated with the specimens in our collection. I applied the method to a bucket where I knew what media I put in it. I went around the collection, kind of went shopping for media, poured it into a bucket, and then I also applied it to those buckets in the collection in the prep lab, where I have a good idea of what specimens are in our collection, but I wasn't exactly sure which of the jars were put into the waste bucket. So this one I would call my unknown. Just to give you some preliminary results, because I'm still sifting through these data, in the bucket where I knew which species I put into the jar, I was able to recover a sequence for 3 species, 2 species, as well as assigning OTs to 2 genera that I knew I put in the bucket. So this is just looking at the very, very top hits from an analysis program called Kraken. And I know that there's lots and lots more data in there, and now it's just up to me to decide where those particular cutoff thresholds are for believing how much of the sequence reads were actually from these species. And even more exciting is that unknown bucket, where I wasn't sure what species went into it, but in just sifting through the top 15 hits on Kraken, I was able to find a whole bunch of species that were very believably in that bucket, both because we have them in our collection and we were actively curating them at the time. So we have lots of different bentwood species that were being processed, as well as central bearded dragon. And 2 species that came up in this analysis that were really cool were the powerful owl and the cagu, because these are specimens that we don't have in spirit. We don't have them in wet collections, but at the time that I was doing the collection of the specimen media, they were processing some of these species in the prep lab to make as skins. And so the DNA that I picked up in the waste bucket is very likely DNA that was floating around in the air, landed in the waste bucket, was fixed in formaldehyde because it contained formaldehyde in the bucket, and then I was able to recover that. And so this is evidence of airborne eDNA floating around in the prep lab that I was able to pick up. So that adds another layer of complexity, knowing that the DNA in the bucket isn't necessarily DNA that came from specimens in the bucket. But I think that that's valuable information to have, and also kind of shows the usability of using a formalin-based media in order to preserve DNA collected in the air. Oh, and over here, you guys are covering it. But we also picked up some sequence for a blowfly, and this is one that is associated with necrotic tissue. So this was likely larvae that were either laid in some of these individuals after they were, before they went into the collection. So sometimes we get roadkill, and that might have been the way that that made it into our sample, or potentially some way through the processing of these specimens. So this all has me optimistically looking into the vault for additional ways of using the DNA that's preserved in these jars, because I think that this would be a really cool way to either recover whole mitochondrial sequences from these specimens without having to do any destructive sampling, which would be really useful if we are sequencing holotypes of individuals. These would be specimens that are preserved singularly by themselves in a jar to serve as the type specimen for a given species. And we don't want to destructive sample these ones because it's the only one that we have and we use it as a reference. But if we can use the gunk in the bottom of the jar to collect genetic information from it, then that would be an invaluable resource for people who are doing lots of other comparative genomics and also eDNA studies around the world. So this is something that I'm actively developing both in our collection as well as looking at in collaboration with the fish collection in Hobart. So I've now been able to provide a method that both reliably sequences specimens preserved in formalin, starting to get evidence that we can get genetic information from the specimen media itself. And now I wanted to take those specimens preserved in formalin a step further and quite literally flip it on its head. So back to the formalin problem. We have all of these reptiles, amphibians, and fish preserved in formaldehyde, which, yes, is annoying if you study these species, but it's particularly vexing because it's these species that, at least amongst vertebrates, are some of the most biodiverse and environmentally sensitive taxa. And if you think about it, each and every single one of these is a little record of how that species was coping, or perhaps not, with the environmental challenges of its day. And it's only these specimens in our collections that are preserved whole. Everything else largely has their internal organs removed and are either preserved as a skin or bone. And so if we're looking at organismal response, the sort of information that bone is going to tell you about, say, gene expression isn't as informative of how a species is perhaps responding to a novel pathogen that attacks their lungs if they don't have their lungs. So these specimens are all preserved with their individual organs. And if there were a way to go in and extract the gene expression information in a tissue-specific way from these specimens, then we would be opening up a whole door to looking at how species respond in a very individualistic way. And the way that we might go about this is by looking at the epigenome. And there's a whole bunch of different marks that we use on top of the DNA that we call epigenomic markers, either DNA methylation, which is one of the most commonly used, also histone modifications, different chemical modifications added to the packaging material in the genome. Also, where those nucleosomes are sitting along the genome can tell you information about the activity of a given gene, as well as potentially RNA and individual proteins in the cells. So if we're able to access this information, then that gives us information about what the genome is actually doing at the time in the context of the environment that that specimen was collected. This would be an amazing approach to apply in collections because we have millions of specimens collected all around the world, and most of that activity was occurring in coinciding with a period of rapid environmental change over the last 150 years or so in response to things like climate change and introduction of novel pests and pathogens. So potentially sitting at our fingertips is over a century's worth of environmental response data, if only we could get to it. So back to formalin. Formalin was thought to damage DNA because it goes in, it cross-links all sorts of proteins and various cellular components to the DNA, and then when you try to get it out, it fragments it beyond belief and you can't extract it. Well, my work has shown that you can get the DNA out. It is still very highly fragmented, but it's still there. It's preserved, and it's largely the extraction method that is what disrupts all the DNA. Formalin isn't seen as a complete barrier to molecular analyses in other fields of biology. In chromatin biology research and immunological assays, we use formalin in order to lock in place all of the cellular machinery in the cell in order to learn information about what it's doing. So approaches like ChIP-seq, Hi-C, FAIR-seq, and MNase-Seq all use formalin in order to gain information. So clearly it's not a complete blocker to molecular analyses. And the subject of my postdoc was to try to adapt some of these methods to applications in museum specimens to see if we can take advantage of historical formalin preservation to gain historical gene expression and epigenetic information. So we've tried a bunch of different assays, and the one that we are just ready to start talking about is our adaptation of micrococcal nuclease sequencing to museum specimens. And what micrococcal nuclease does is it's an enzyme that goes in and it chops on either side of the nucleosome and then eats away at the intervening DNA in between them. And so you get an idea of where individual nucleosomes are sitting in the genome. To develop this method, I didn't want to go and start using precious museum samples, so I put together a mock museum collection, which consisted of both inbred lab mice as well as some wild-caught Australian mice. We processed the samples the same way we do as anything else that enters the collection. We took a fresh liver tissue sample, popped it in the -80 to do downstream RNA-seq as well as a micrococcal nuclease treatment of fresh tissue. We also put the remaining carcass into formalin, fixed it for 3 days, and then washed it and put it into ethanol. And by the time that we got around to processing these samples for sequencing, they'd been sitting in the collection for between 2 and 5 years. So these were properly archival and much, much heavier fixed than, say, something you would see in a histological lab, which might be fixed for 15 minutes. So these are really properly heavily fixed. This is quite complicated here. It's many, many steps, but this is the method that we ultimately landed on that was allowing us to recover historical information from these kind of specimens. We've just put this up on bioRxiv, and again, there's a QR code if you're interested in reading the nitty-gritty, but the bulk of it is just processing the tissue appropriately so that we can get to the chromatin and allow the micrococcal nuclease to do its work. That's what all the little steps in there do. And then we just do really, really heavy sequencing of the resulting DNA fragments and map that, and then do the same kind of trimming-free alignment that we did with our whole genome sequencing approach. And then we apply a enrichment analysis program called DANPOS, which looks for both enrichment as well as depletion signals within those alignments. And I'll just give you a taste of what the signal looks like after applying this archival micrococcal nuclease assay. So these are both just whole genome sequencing between the archival sequence data on the bottom and that on the top. And you can see that the cover is pretty similar between fresh and archival tissues, which in and of itself is pretty exciting. But once we apply the micrococcal nuclease treatment and start eating away at that intervening DNA, we can start to see that differences emerge in the coverage patterns between the fresh and the archival tissue. And when we apply that analysis of DANPOS, looking at the difference between the micrococcal nuclease treatment and a input control, we start to see that those differences between the fresh and the archival samples become even more exacerbated. In the fresh samples, what we typically see is a positive signal, so enrichment that is associated where the nucleosomes would be sitting in that particular stretch of DNA. So we see a signal of positive of peak gain. On the flip side, in archival samples, rather than seeing a signal of peak gain, we see a signal of peak loss. And this is, although mirrored, it allows us to look at differences in sequence cover that we can associate with gene expression. So that was looking at one gene. Looking across the entire genome, on the left-hand side, this is looking where peak gains have been detected at the transcription start site, lining up all the genes across the genome there. When you see a signal of enrichment in response to micrococcal nuclease treatment, you see this kind of tornado effect. So in peak gains, we see that tornado effect in the fresh tissues, but not in the archival tissues. However, when we look at peak losses across the genome, we don't see the tornado in fresh signals, but we do see it consistently in archival signals. So the signal that we're getting from micrococcal nuclease treatment of archival tissues is the mirror image of fresh tissues. And the signal isn't completely lost because of heavy formalin fixation. It's just inverted. We had to look at it differently. So That paper is going to be coming up pretty soon, which opens up this whole idea of using historical gene expression information to start tracking environmental change. And that's really, really exciting. But that was on mice. And what we want to do is actually look at some species that might be of conservation concern or are otherwise relevant to our local environments. So in addition to putting it to work, to testing it in our mouse samples, I also wanted to apply it to some actual archival samples. And for this component, I teamed up with some researchers at the University of Queensland, Celine Frere and Nicola Jackson. Celine and Nicola, they study eastern water dragons, and they're looking at the response, the adaptive response of this species to urbanization in the Brisbane area. And they've collected a whole heap of modern data, including behavioral, morphological, genetic differentiation data, as well as the prevalence of a fungal pathogen that is being pretty devastating for this species. And their system is pretty cool because they have this whole gradient of urbanization across the Brisbane area, which includes populations that have been in urbanized areas for longer or relatively recently. So they can look at the adaptation in real time for these water dragons. But to look at how those populations have changed through time, they needed to get access to historical information. And for that, they came to us. And in the first instance, we sent Nicola into the Queensland Museum to find some specimens to do some testing on. She took our specimen vetting system with her and applied it to a bunch of jars in the collection and found some that we thought would work really well for sequencing. And these included 5 specimens, 4 males, 1 female, and the oldest of which was collected in 1905. We then put that through both our archival genomic sequencing as well as archival micrococcal nuclease assays to look for both genetic and gene expression information. And these are those sort of tunnel plots, those funnel plots that I was showing before. And what we're looking at is the 5 individuals and whether or not we saw enrichment at the transcription start sites in response to our micrococcal nuclease treatment. And the first thing that we see is that there is a, albeit weaker, but consistent signal of expression across samples collected over the last 100 years. And what's really cool is that the oldest sample is also showing a signal. So there doesn't seem to be an effective age of whether or not the assay works, which is consistent with our results with genome sequencing, in that there doesn't seem to be any kind of effective age. So long as it was preserved well in the first instance, it's kind of just sits there waiting for you until you're ready. And the second thing that we notice is that in combining both the genomic sequencing and the micrococcal nuclease data, we get really good cover of the genome, upwards of 14x in some of the more recent samples. And this is enough to call SNPs across an entire genome for historical sampling. So this allows you to do phylogenetic analysis in addition to gene expression information analyses for specimens up to over 120 years old. So that's all well and good, but what can we actually do with this? Can we pull out biologically relevant information from these kind of specimens? Now, we do only have 5 samples here, so we do have to, kind of take this with a grain of salt. But what I did was take the micrococcal nuclease sequence patterns across the entire genome and just look for pairwise differences between individuals using a Pearson correlation analysis, popped them into a PCA. And the first thing we notice is that all of the samples collected, all of the male samples collected in the city kind of cluster down in the bottom. Oh, wait, hold on, I have arrows here. There we go. Okay, so we have a cluster of males collected in the city, which are all together despite over 100-year difference between them. We have one individual female that's up in her own little world, and then we have another male that was collected in the natural population. When we then look at PC2 and PC3, we see that the influence of sex drops away. So that one female who is off in her own little world then clusters more tightly with the male that was collected also in natural habitat. And so we see that in addition to there being an influence of sex on epigenetic signature, we also see a signature of habitat differentiation. And these are both things that you would expect, which is why it's really, really important to do sex matching if you're going to do any epigenetic analyses. So while this doesn't give us any direct insight into this particular species, it does give us guidance on how to conduct future sampling, and it does give us an inclination that the information that we're getting using this assay does reflect what we would expect. The next step that we are engaging in is to create a fundamental data set that will enable other researchers to do smaller scale epigenetic research and then put that into the context of what they would expect to see across the genome, given our much better understanding of how the genome responds in a whole bunch of different taxa. So we're going to be sequencing 1,000 epigenomes collected over 100 years for 10 different taxa. And this will form a fundamental data set for reptiles, mammals, as well as amphibians, in order to then put that into context and understand how different species are changing through time. So this is something that we are actively looking for partners to kind of select our species, because we want to make sure that the species that we select are both representative of the kind of taxa that people want to see information for, and also that will be immediately usable for someone's research. So if you're particularly interested in this, hit me up and we can see if we can potentially look at a partnership to include one of your species in this kind of work. So the last thing that I'm going to touch on is to then just give an overview of the sort of work that I'm doing now at the CSIRO. All of that work was mostly to do with my postdoctoral work. I am seeing it through, but I'm now been kind of tasked with leading an effort to do an airborne eDNA study at the CSIRO. And so I'm coordinating a whole bunch of different researchers across different units. And we're going to be trying to push this technology forward in order to develop and deliver new capabilities. So just a quick little touch on why eDNA. I like to think of eDNA a bit like glitter. If you have children like I do, you know that they come home with artwork and glitter ends up everywhere. It's no different for environmental DNA. We have been able to use eDNA in all sorts of different ecosystems, from those that are really, really difficult to access to ones where we are very sensitive environments. We don't necessarily want to go tromping through them. We can use DNA shed by a variety of different organisms to track their movement through different ecotypes and to survey what species are there. And so this sort of technology has been really well developed in aquatic systems, and it's only more recently that we're starting to think more and more about terrestrial eDNA. But coming from a conservation genetics background, we didn't used to call it terrestrial eDNA, we used to call it non-invasive sampling. We do lots of eDNA collection from scats, from feathers, from you name it. If we wanted to pick up traces of individuals, we're quite creative in the ways that we collect DNA. We just didn't call it eDNA. And now that we're starting to, revisit what we're calling those kind of samples, we're being even more creative with the sources that we're getting eDNA from. And in the last few years, there's been a couple studies literally plucking DNA out of the air in order to track a whole bunch of broad taxa. That technology, I think, is really ready to take off, but we want to take a moment to think about how we can take advantage of the vast amount of information that has come from using eDNA in aquatic systems before we get too excited and apply it inappropriately to airborne eDNA. So there's lots of advantages to using eDNA in aquatic systems. It's non-invasive, so you don't have to go out and do electrofishing to detect fish. It's very sensitive. You can, just from a small amount of water collected, you can get lots of different taxa. And it's also comprehensive. It's something that can be used to simultaneously assay both animals and plants, as well as fungi and microbes. And to that regard, it's very cost effective because you don't need bespoke methods for detecting a whole bunch of different species if you can do it all from one little pot of DNA. But we can't talk about the advantages of eDNA without also acknowledging its limitations. It's largely viewed as not quantitative. You don't know how many fish are in the water, and the fish that you are able to detect, or whatever taxa that you're looking at, it's entirely dependent on the reference database that you're using. And those reference databases have biases in them, both in terms of which species are in them and how what quality the DNA sequences are in those databases. There's also a bazillion questions around DNA persistence. If you pick up a particular species, does that mean that it was there this morning, or does it mean that it was there 30 years ago? That it isn't always clear. And of course, the expense of eDNA is in both columns, because while it can be very cost effective to simultaneously assay a whole bunch of different taxa, if you're particularly a group that doesn't do molecular work, this can be something that is extremely expensive for you to try to kick off an eDNA program. In aquatic eDNA, they're starting to break down the barriers on these different limitations, and it's just acknowledging what eDNA can do, where it can be appropriate, and then really focusing on the ways that we can apply it so that it's most useful. In that quantitative question, I think it's pretty well understood that no, it doesn't help you count things, but we are seeing more and more papers which are associating the amount of DNA that you have, or the number of hits that you get from a different sampling site, to estimate at least abundance or biomass in some way. And you can have a pretty good sense of being able to tell there's more of that thing than there used to be, or there's more, we're getting more hits, something's changed. So while it's not entirely quantitative, it is giving us some information about population changes. The other question is about those reference databases. There seem to be a bazillion different reference database projects up and running now. And at the International Congress of Genetics, I heard about a whole bunch of new efforts that are going on all around the world. I will just highlight one that we have at the CSIRO. We are building a national biodiversity DNA library, and our aim is essentially to go out and sequence one of everything. And that sequence is also tied to a voucher specimen in a collection, so that you can always go back and taxonomically identify the species from a very well curated database. And so that's going to be a step better than using NCBI, which is a collection of sequences that kind of anybody wants to put in there. And so hopefully in a couple of years, that reference database question isn't going to be as such much of an issue. Around the question of DNA persistence, there's a whole bunch of studies coming out around the ecology of eDNA. And one that I thought was pretty cool was looking at the relative abundance of eDNA versus eRNA. And since RNA is a bit more fragile, looking at its stability through an ecosystem in relation to DNA can start to give you information and that can allow you to predict how old that DNA, or how long ago it was shed. So again, we might not be able to say that it was shed 17 hours ago, but you might be able to get a good sense of, was it shed today, or was it shed quite a long time ago, which can be really helpful for wildlife managers. And the last question is that expense. It is still very expensive for someone to kickstart an eDNA program in their lab, even if they have molecular experience. But we're starting to see service providers like WilderLab, who you might have heard of over in New Zealand, and these sorts of service providers are going to be taking it out of the hands of individual labs to do eDNA research, because they'll send you a kit, you do your sampling, you send it back, and they give you a whole list of species that they found in your area. And I think this is really where the field is going to take off, because now it's not going to be something that each lab has to do individually. Because they can do things in bulk, we're going to see a lot more work done. So I'm really excited to see these groups start to take off. And that's where I think the next stage is going to be in airborne eDNA, because air, like water, it's a fluid, stuff moves around, we can get DNA out of it. We just need to make sure that we can do it effectively and reliably. And some of the really exciting studies have come out in the last couple of years on airborne eDNA. One, which was here in Current Biology last year, they put out an air sampler in a zoo and pretty much detected the distance. So the further away from something that you are, the less likely you are to pick it up, as well as sort of the more biomass there is. It makes it, more likely to pick up that species. Another really cool study was using air quality monitors to do eDNA sampling. There's these systems that are already in place going out and telling you all about the pollution in your area. And they took the filters from those, did an eDNA approach, and they were able to recover over 180 different species associated with the local environment. So this is showing that the taxon you're able to pick up species, even ones that you wouldn't necessarily think you could, such as amphibians, and that this method might be really, really useful for assaying biodiversity. And we also have these existing infrastructure networks of air quality monitors all across different countries. So like all around Europe, all around North America, we have these systems in place that we can utilize for dual purposes. But that doesn't really help us if we work in areas which don't have these really extensive networks. How do we survey our really diverse ecosystems, especially in corners of the world where we don't have large swaths that aren't populated? How do we go out and do eDNA monitoring here? And that's where I'm focusing my research over the next few years, is on making AirDNA technology accessible to everyone. I want to build a platform that has large taxonomic breadth, so it doesn't matter what species that you're working on. I want something that preserves DNA long term, because we have a lot of groups that go out, they do surveying once every 3 months, potentially once every 6 months, and they're not going to go back the next day to do their survey, to collect their devices. I want something that works for them. And that is also compact and lightweight, so they're not trudging around big equipment, something that's affordable, so almost anyone can do it, and something that's not non-technical, so it doesn't require understanding of sterile technique or molecular DNA analysis in order to deploy devices and understand the data that come out of them. I'm testing a variety of different trap designs, both active ones that pump air, as well as things that collect DNA passively. And my preliminary results show that both are effective. They work really, really well. So some big contraption that pumps DNA is probably just as good as something that essentially you're waving a tissue around in the air. The work that I have in progress right now is on pushing the boundaries of where eDNA can go and when it breaks down. So we'll be testing different temperature regimes, humidity, longevity, how long does it last? Does wind matter? Does it matter where you're putting the device, and which species can you detect with it and which can't you detect with it? I'm partnering with some organizations here in Australia. So I've got a project up and running with the New South Wales government, the Department of Planning and Environment, to put out traps in coordination with their traditional monitoring methods to look for different endangered species of microbats as well as rodents. And I'm also working with the National Territory, the Northern Territory National Resource Management Group, to put out our eDNA devices alongside bioacoustic devices to look for the white-throated grass wren, as well as the co-occurrence of domestic cats. So this will be a really cool opportunity to test the platform in very different environments, as well as in looking for different species and whether or not it's going to work in the hands of ecologists, which I'm really excited to see. And all of this is going to be done in the new state-of-the-art collections building that is just up the road from me and is still a big hole. But as of next year, we're going to have a state-of-the-art trace facility right next to our collections. So we're going to be a hub for this sort of work going forward. So that was a bit of a lot of stuff that I've been doing over the last couple of years, and it involved a whole lot of people, both here at the CSIRO, UQ, ANU, and that should say University of Canberra. They're not ANU, both of them. But yeah, it's been a joint effort, and it's been funded by Bioplatforms Australia, and I've got some of my contact details on there if you want to ask me questions via email or hit me up on Twitter. But otherwise, you guys can pester me with questions until your heart delight.
Awesome. That was great. I clap on behalf of everybody. Hope you heard that.
Yeah.
Cool. So we'll go into, you've stopped sharing. So we'll go into feedback and questions from the audience. Does anybody have any questions? Okay, well, I can start off. I was really interested in lots of different elements of that, and I wondered, when you were sort of talking in your really first part of the talk, what would your advice be to people that are trying to do whole genome resequencing from old samples that are like up to over that sort of 40-year barrier that you mentioned. Because I think we can by now get, small quantities of DNA even, from these samples. In fact, I've sort of worked on samples before and haven't really been that limited in concentration, but it's the kind of breadth of the genome that the sample's representing, I guess. So do you think there's hope that we will get to a point where we can get decent coverage out of museum samples at that whole genome level?
Yeah, I do. And I put the 40-year marker in the paper because we had so few individuals that we had sampled from prior to that. And so we just wanted to mention that age might be a factor. But since publishing that, we've done a bunch more sampling, in particular in the water dragons. And I don't think age matters when it comes to formalin-preserved specimens, because once the formalin preservation is done, it just kind of sits there, which is different than something like pinned insects, which are just going to be continually degrading over time. So it's a bit of a different story. And where I'm reporting that we're getting, 11x cover, 3x cover, it's because these are species with like 1.5 gig genomes. And it just depends on how much money you want to pump into sequencing them as to whether or not you're going to get really high cover. And just recently this year, the cost of short read sequencing has again dropped because there's new sequencing platforms that can pump out tenfold more sequencing reads than they were able to do previously. So short read technology still relevant, and it just kind of depends on how badly you want that genome. And the other thing is using a mapping approach that is going to get as much information as you can. So we don't do any kind of trimming of our reads at all. We map it using a program called K-Align. And the other thing we do is that we use a library prep kit that suppresses any primer dimer and also has unique molecular identifiers. So we're able to, in our initial paper in Molecular Ecology Resources, we were losing 60% of the data because it wasn't mapping. I think most of that was primer dimer. And then in the second round, a lot of it was over amplification because we had so little to begin with that we hadn't really honed that in. But now I'm getting upwards of like 70% of our stuff mapping and retained after deduplication. So now we're getting to the point where we can, from almost any specimen, get whole genome sequencing cover, at least in formalin-preserved stuff. So.
Oh, that's really exciting.
And I think those sort of methods can be applied to anything, so long as yeah.
Yeah, cool. There's a question from Georgia. Since she typed it, I'll read it for you. It says, "Very cool work. Thank you for sharing. How long of amplicons have you been able to get from formalin-fixed DNA?"
Small. It's the average fragments are under 100 bases. You might have a random longer one in there, and so maybe you'd be lucky if you're targeting, fragments that are 150 or 200 bases, you might be lucky. But in our Bucket-Seq approach, I want to say they were 80, one of them was 80 base pairs and one of them was like 110. So you do really need to target really small regions, but if you have a region of interest, you just tile them. And again, it just depends on how badly you want that sequence data, because it's not going to work if you're targeting 300 and 400 base pair amplicons. That's something that we still struggle with, where people will try our method. They'll try an amplicon that's way too big and then they'll tell us it doesn't work. And I'm like, well, you're never going to get your 500 base pair amplicon. It's just not going to work. And we are also trialing doing reduced representation sequencing on the output from our extraction. So we've got samples over at DART here in Canberra to see whether or not that will work. There is a lot of evidence to show that bait capture works really well with these samples. And so that would become a more affordable way of doing population genetics on these kind of samples.
I have another question. just a quick one, maybe to wrap us up. So you mentioned at the beginning that you were talking about how there's there is starting to be consideration in Australia and also around the world about data sovereignty aspects of all of these different types of data. In the environmental DNA program that you're developing at the moment, and also the museum work where people, have historically deposited samples, well before any consideration of these sorts of issues. How's your group kind of working towards addressing these issues, or how do you foresee this working well in the future?
So 2 questions there. The curation side of it, it's honestly something that we are struggling with. It's something that we are just now starting to more comfortably even have conversations within our own collection about it. And it's something that we are working with other institutions internationally to learn from and figure out how to do this really well. There's a workshop coming up here in Canberra, hosted at the ANU, and it's going to be something that's involving researchers and curators, because researchers are doing, collections. They have historical collections that they've inherited from different people. And just understanding the breadth of the problem and then trying to figure out how to do something with. I'm not saying we're doing it well. And it's, I think, something that we need to learn how to have uncomfortable conversations about more and more so, because even I've had researchers who want to ask me like, oh, I want to access specimens in the collection. What do we do about data sovereignty? And I'm like, I'm not sure. Thank you for asking. Let's dig in this together, because it might be something very specific to those specimens that we don't have a protocol for. So unfortunately, I don't have a really good response that I'm happy with on the collections end of it. On the eDNA side of it, something I'm working with from the very beginning with airborne eDNA is, because these studies will be conducted in collaboration with traditional owners, we're going to be doing having discussions on the first hand of what it means, what this information is going to be, so that we can very, very openly say, the DNA that we're collecting, it might be yours, it might be your relatives. And what does that mean for you? How does that fit into the information that you want to get out of it? Because we do have the ability to do like blocking of human DNA. Do you not want that? Are there particular species in the area that you're concerned about that you want information on, in which case we'll target that? Or are there species that you would rather us not provide information about, because that would, open you up to people knowing where sensitive taxa are. So from the very beginning, we're consulting and also involving the traditional owners to make sure that it's a co-design process. And we won't be engaging in any kind of sampling that isn't a co-design from the very beginning. And that's something that I'm trying to push really hard. And it's something that I'm also trying to kickstart a bit of a thought, like a thought experiment on what is eDNA, to instigate a conversation around the world. And so I met a researcher, Dawn Lewis, at ICG just a couple of weeks ago, and she does research on paleogenomics, and she herself is an Indigenous woman, and I want to get her insight on what it might mean to translate that kind of work to eDNA, because I think there's a lot we can learn from the ancient DNA space, who's grappled with these kind of questions already, that we can also translate to eDNA and just not reinvent the wheel and make sure that we're all communicating really well. But a very active area of discussion and research.
Yeah, cool. Well, I guess we've got 1 minute left on our timeline, so I'll just see if anybody else has any quick last minute questions. Okay. All right. Well, thank you again. That was a really excellent seminar, and lots of interesting things covered. So you've given us lots of food for thought. I'll stop recording now, and